So anyhow, just try not to get too far behind. And if you do, just let me know. I'll try to help you, give you some advice. All right. So homeostasis. Um, your OpenStax book briefly talks about it in chapter 1. It talks about it in a few other chapters kind of throughout. So I wanted to kind of condense this idea of homeostasis and talk about it a little bit, because it's really kind of the foundation of why you're here and why you do what you do. It ties together all of our body systems. It ties together all of this chemistry and cellular level stuff that we're going to talk about a little bit for the next few weeks. Next semester, you're going to spend basically in advance the entire 14, 15 weeks talking about homeostasis and analyzing chemicals and cellular levels and why homeostasis isn't maybe happening and how do we get it to happen? It's kind of what we do in the clinic all day. So we're going to define homeostasis. We'll look at some of the main mechanisms involved with homeostasis. In general, it's the consistency and those levels that we maintain that keep us in balance. So a lot of times homeostasis is a way of saying balance. It's kind of like the opposite term for disease or pathology. When someone has a disease or a disorder, an aspect of that individual, which it could be an organ, it could be systemic, the entire body systems put together are causing the individual to be in some sort of some state of disease or have multitude of disorders. Homeostasis is the opposite of that. Homeostasis is where we should be most of the time, if not all of the time. There are ranges within homeostasis. So we generally think of homeostasis simply as your body's ability to maintain balance. Okay, and some of the vital signs that you're going to be taking in the clinic: body temperature, heart rate, blood pressure, respiration rate. These are all types of features that the body is going to perform and utilize to tell us that we're in homeostasis. If we're not at 98.6, we're above that, we're maybe in a fever state, that's a signal that we're not in homeostasis. And it's also a signal that something is wrong. There could be an infection. There could be stress. There could be any number of reasons why our body temperature has gone up. If blood pressure, right? Blood pressure goes up when we're under pain, when we're under stress. When we have certain blockages, we may see blood pressure alterations. Same with heart rate. So anyway, kind of maintaining balance is homeostasis. So again, temperature regulation is a big one. Maintaining that 98.6 degree Fahrenheit. And again, there's a small range of acceptable numbers for all of these. Regulating carbon dioxide levels in the blood. We don't really, most of these, by the way, are involuntary. They're outside of your thought process. These homeostatic mechanisms are occurring because we want you to stay alive and because you need to be alive. So they're basically survival mechanisms that keep us going. And again, we don't really have to think about them that terribly often. So 98.6 for temperature. Carbon dioxide is a waste gas that you exhale when you breathe. It's a waste gas that comes about from cellular respiration or from cells, in essence, breathing or converting energy. So carbon dioxide is something we want to be able to eliminate. When there's a buildup of too much carbon dioxide, it can alter what we call the pH balance. And you've probably heard pH balance at some point. So we won't get into that too much now. We'll get into pH later, but carbon dioxide levels are regulated by our respiratory levels primarily. And then certainly regulation of blood glucose is another example. we've all heard of diabetes. We've probably all heard of insulin as well. And so the body has a constant regulatory mechanism that is maintaining blood glucose levels. And what blood glucose does, or what glucose does, is it enables our cells to be able to convert the glucose into energy. So anyway, homeostasis constantly occurring, constantly being regulated and monitored. This little illustration is showing the internal environment and the external environment kind of coming together, and what's going on, and a variety of different systems working together. I think really the goal of understanding the human body, especially, you know, after last week, you guys looked at body organization, you looked at the chemical level, and then the cellular level, and the tissue level, and the organ level, and ultimately the organ systems, which is how we're going to go about studying A&P this semester, is system by system. Now, you can get away from this idea of everything being together and working intrinsically with one another to attain and maintain homeostasis. You can sometimes get away from that concept, which you won't in here because I'll remind you of it throughout. But because we do compartmentalize all of these body systems, we will just look at bones. We'll just look at muscles. We'll just look at nerves. That doesn't mean that nerves don't affect muscles, and muscles don't affect bone, and so on. So all of these body systems, organ systems, and organs are intrinsically linked. And that's kind of what this diagram, I think, is trying to show, is that we bring in nutrients, water, we bring in oxygen from the external environment to the internal environment, and that enables ultimately our cells to work with all of these ingredients that we've provided them with, which were delivered to the cells via the cardiovascular system. So we have this cardiovascular system of the heart and all of these blood vessels that's distributing these nutrients and as well as taking away the waste. So we see the digestive system and the respiratory system are the primary systems for bringing in the materials for the blood to deliver those materials ultimately to the cells. And then the cells are going to poop and pee and eliminate waste. Cells are also going to give us some of that, some energy molecules. They're going to provide us with some protein structures and some other chemical enzymes. But there's going to be some waste involved, and that waste is going to leave the cellular level, go into the bloodstream, and be eliminated through the urinary system as well as the digestive system. So groceries in, garbage out. And we see the respiratory system has a dual groceries in, garbage out in the same pathway. So we inhale the oxygen, we exhale the carbon dioxide. With the digestive tract, we've got opposite ends. We bring in through the mouth nutrients and water and vitamins, minerals, and we eliminate unabsorbed matter and waste through the other end. Okay, and then we have the urinary system, which is completely internal until we're talking about elimination. Once we're ready to eliminate some excess salts, excess water, what we call nitrogenous waste, such as ammonia and urea, that's going to leave the body via that urinary system. So again, we see 1, 2, 3, 4 body systems represented right on this slide: respiratory, digestive, cardiovascular, and urinary. The bulk of the second half of the semester is going to be going back and looking at these systems in a little bit more detail. So anyway, I'm not going to belabor the point. How do we maintain homeostasis? In essence, this goes back to some of the basics that you maybe learned in psychology at some point regarding some sort of a stimulus leading to a response, right? I touch something hot, the stimulus is, that is hot. That signal is going to travel somewhere to the brain. The brain is then going to send a signal out to my muscles saying, you need to remove your hand from whatever it is you're touching that's hot. So the response is to remove the hand, to go against or go away from that original stimulus. So again, homeostasis is all about stimulus and a response. And in between, that stimulus is going to get sent to what we call a control or an integration center. You guys know it as your brain. And then that control center, the brain, is going to send out or elicit some sort of a response. So this is simply stimulus comes in, response goes out. And in the middle, we have this integration or control center. So the basic components of homeostasis involve a sensory mechanism. For us, it's going to be in the form of nerves, right? Nerves are going to be the sensory mechanisms, and those sensory mechanisms are going to be located within the skin. They're located within the body as well, and some of the membranous structures, certainly in the mouth. Okay, so a sensory mechanism, so some sort of a sensory nerve is going to be kind of monitoring the environment, picking up temperature changes, picking up pressure changes, picking up whether something is sharp, sensory changes regarding like tactile stuff, sharp, fuzzy, pokey, those types of things. So those are more external. Internally, we also have sensory receptors that are going to pick up whether there's some sort of inflammation, right? Like if your muscles are sore, or if you have kind of like a gas pain or something in your abdominal area, those pains are being picked up by smooth muscle, skeletal muscle inside of the body, and delivering that pain signal or pressure signal, whatever that the sensory is, that's being picked up by nerves and then delivered to the brain or the control center. At that point, depending on what the stimulus is, typically if it's a temperature issue, a pain issue, a pressure issue, inflammation issue, typically the brain wants to elicit or send a response that's going to cause an effect. That's where you get the E for effect. So we should get some sort of an effect that is delivered or sent out, some sort of a response sent by the brain. And this is also going to be some sort of a nerve pathway. So you're going to have an opposite pathway of nerve or electrical flow going away from the brain that's going to deliver the response message. And typically the response message or the nerve signaling is going to go to a skeletal muscle, a smooth muscle, or a cardiac muscle, typically, or a gland of some sort. So the effector mechanism is usually going to be a nerve heading to a muscle or a gland of some sort. Okay, so cause and effect, stimulus, response. Okay, and in between, it's the brain that's controlling that, receiving, as well as sending out signals. It also stores memory or information based on whatever information has been coming in. Okay, that effector, that response mechanism is also called feedback. Okay, so we'll look at feedback in just a second as well. We call them feedback loops. Okay. I also wanted to mention set points. Set point is that homeostatic norm that an individual has, that we all have, that 98.6. That's our set point for internal body temperature. 120/80 is our set point for blood pressure. 60 or 80 beats per minute. That's our set point for heart rate. Okay, so set point is your normal numbers. Now, set points can be out of range for some folks who are maybe going through chronic issues. So blood pressure, for instance, we may have a patient who comes in and we take their blood pressure and it's 140 over 90. That's too high. That's 120 over 80 is normal. Theirs is 140 over 90. So we may say, oh, they have high blood pressure. Maybe they do. Maybe it's acute, like meaning it just happened, like they had to take some stairs, they were running late, something, they're stressed out about doctor visits. So those things could like, right, a stressor could raise your blood pressure. So we want to find out if Mr. Smith, who came into the clinic with a 140 over 90 blood pressure reading, we want to find out if that 140 over 90 reading is a fluke, it's just a in the moment issue, what we call acute, or does Mr. Smith have a new set point? Has he been in a situation, whatever it might be, stress or pain or some sort of disease or disorder or pathogenic infection, has he been undergoing some sort of change that his blood pressure is at 140 over 90, but that's where it is all the time? So in other words, that's Mr. Smith's new set point. That's not good, right? We don't say, oh, well, that's Mr. Smith's set point. It's good enough. It's his. He owns it. He's built up to that. So let's let him have it. No, we don't want to approach it that way. First of all, we want to find out, is 140 over 90 the new set point? So we would have Mr. Smith do his own blood pressure, kind of keep a log at home for a couple of days, maybe for a week or so, have him do his blood pressure 3 or 4 or 5 times a day, maybe once a day, maybe twice a day. But either way, we're going to have him do his blood pressure and log it. Nowadays, they do a lot of virtual logging. And so the physician in the clinic, we can see in real time what Mr. Smith's blood pressure is. So from those numbers that he's logged, we can then, maybe he leaves the doctor, he goes home and he does his blood pressure. When he gets home, he's 120 over 80. Goes to bed, wakes up, he's around 118 over 78. Checks it at noon, he's 120 over 80. So the pattern for Mr. Smith when he's at home and when he's doing his own thing is, oh, he's got normal blood pressure. So the 140 over 90 at the clinic was an anomaly. He was stressed out. It could be an indicator of an acute infection or something as well. But again, if he goes home and it's 140 over 90 every single time he takes it, or it's high like that every single time he takes it, he has a new set point. So set points, my point is, set points can become altered into ranges that we don't like, what we call abnormal ranges. Okay, so set point doesn't necessarily mean homeostasis. Set point means that an individual's numbers, when we measure vital signs, may be outside of normal ranges as a set point, as a consistent chronic set point or number. So anyway, I wanted to mention that too. And then feedback, that feedback that we get is, again, effector. That is the response. Okay, so think of feedback as a response. Most responses are elicited or sent out by the integrating or control center, by the brain, to oppose the stimulus. So if the stimulus is, that's really hot, the response needs to be, pull your hand away from what's really hot. So I'm opposing the stimulus. Okay, the stimulus is, this is really hot. I want to oppose what's really hot. So I could dump ice or cold water on it, or pull it away, or both. Okay, so we call that type of feedback response negative feedback. So almost everything that is going on in our lives is negative feedback. It is your brain opposing a stimulus that is, picked up by the sensory, that is not supposed to be happening. So if the sensory is blood pressure is going up, the brain will work to send a response to get the blood pressure to come back down. It may send a response to slow the heart rate, to dilate some vessels, to thin out your blood. Okay. If the response is we don't have enough blood sugar, or if the stimulus is we don't have enough blood sugar, the response by the brain is going to be, let's get some stored glucose going, let's get some hunger centers going, let's get some sugar glucose back into the bloodstream. Okay, so stimulus response. That response almost always opposes a stimulus. Not always, almost always. And we call that negative feedback. Okay, it's inhibitory and opposes the stimulus. Okay, it's there to stabilize the variables, bring everything back to homeostasis. Okay, so it produces an opposite change, and it's what's responsible for maintaining homeostasis. It's something as simple as you walk into a bright room, or you're inside and you go outside on a bright sunny day, which we don't have today. But you go in from inside to an outside where it's really bright and sunny. You don't have sunglasses on. The stimulus is coming into your eyes and through the pupils, that the lighting is really bright. Okay, so we want to oppose the stimulus. We want to oppose the brightness. So we're going to get negative feedback. What that means then, the brain gets the signal, it's way too bright, right? The response is going to be, let's shrink the pupils. So your physical response, the feedback that's coming from the brain to the muscles that control pupil diameter, are going to cause the pupils to shrink, because what the pupils do is they allow light in. No different than if I'm going from a bright room into a really dark room. The stimulus is, now I'm entering into a really dark room. The response needs to oppose the darkness. So we're going to get pupils dilating or getting larger, because a larger pupil can allow any light that may be available to go to the brain. So anyway, negative feedback, that's kind of the consistent theme in everything we do. We do have positive feedback. Positive feedback is going to enhance the stimulus. We see this particularly with childbirth. So we generally associate positive feedback with birthing as well as breastfeeding. The stimulus that the mother is going to get when the baby is at 36 to 40 weeks is that there's a large item in my uterus. So the stimulus is large item in uterus. I want to contract, right? So the uterus starts contracting. That contraction is to stimulate and enhance the stimulus of, okay, this is in here. We need to get it out of here. We need to enhance the uterine contraction. So as the baby's getting bigger, the stimulus comes up and the uterus it starts contracting. We don't want to oppose the stimulus. We don't want to stop the uterus from contracting. We want to enhance the uterine contractions. So that's positive feedback, enhancing the stimulus. Stimulus is, uterus is contracting. The response is, great, let's contract it more, versus, the uterus is spasming, it's contracting. What are we going to do? Oh my gosh, let's stop that muscle from contracting. That's negative feedback. We don't want that. We want to enhance the stimulus. So anyway, that's what's going on there. That's about it. There's a couple other things real briefly with homeostatic control. It does happen inside of the cells. So cells of the body have the ability to create enzymes. They have the ability to alter their levels of needs. So anyway, intra means inside of, so inside the cells. Okay, and then we have intrinsic, which is sort of like intracellular, but we are going to be in the body, but not inside of the cells. So we're going to be just outside of the cells. So we're going to see intrinsic control or regulation occurring between tissues. This basically is a communication system. So we have communication inside of cells, we have communication between cells and tissues and organs, and then certainly organs communicate with one another. So that's all. That's basically, this little discussion is simply to, again, show you that everything is intrinsically connected and that we have this continual monitoring of all stimuli, whether the stimuli are external or the stimuli are internal. We're going to get some sort of response. In the clinic, what we're doing is analyzing all of those vital signs to be able to understand whether the individual is healthy, which means they're in homeostasis, whether they might have an acute issue, just an issue that popped up and that we could treat, and they'll get better, the prognosis or outcome or outlook looks good. Hey, we can determine that. So they might be out of homeostasis just a little bit, not enough to have altered their set points. To be the third issue that we're looking for when we do vital signs is this individual suffering from some sort of a chronic disease or a larger scale pathology. All right. So anyway, that's homeostasis. Your case study assignment, your lab assignment this week that we'll look at a little bit on Thursday, goes back through and looks at some of that, and in particular, as it relates to body temperature and hydration, so, and heat stroke. All right, so let's move into the chemistry of life. There's just a handful of items that we need to get through with this. There are some of you have had chemistry in the past. Some of you in your biology class maybe remember some of this. But I'm not here to scare anybody. I'm not here to demand that you memorize a whole bunch of chemical terminology. I really just need you to understand what is an atom, what is an element, what's a molecule, compound. A couple of these terms you've probably heard before, but maybe we're unsure what they meant. So remember, the chemical level and that atomic level, that's where we're going to start with analyzing the body's different organizational structures. So we're going to be at the smallest structure, looking at some of the chemistry that's involved with the body. So anyhow, we'll look at atoms, molecules, compounds a little bit to give you a couple of examples. Isotopes. We'll look at a couple different types of chemical bonding, and again, how they relate to compounds or molecules. We'll look at inorganic compounds. We'll look at organic compounds. We'll look at water, the importance of water, and then talk a little bit about enzymes and their role. So sounds like a lot, but it really isn't. Again, that outline that you have goes into a little bit more detail with a few items regarding the organic molecules, which I'll talk about in just a second. But let's get to it. The elements, we'll look at elements and compounds. So matter, anything that has mass or takes up space. And again, this could be on a micro scale. We look at matter. In the past, we've generally looked at solids, liquids, and gases. Now we see plasma as a form of matter as well. There's a fifth form of matter that's being thrown around in addition to those. But generally, when we look at the body, what we're going to be focused on are solids, liquids, and gases. Those are going to be the three primary forms of matter that we deal with. There are elements. You remember the periodic table. So we'll look at the periodic table. There are, 120 or so elements. And out of that 120 or so, there are 26 of those are found within the human body. And out of that 26, about 95 or more percent of, so 4 elements out of this 26, 4 elements make up about almost 100% of the body. So that makes things nice, right? We look at a periodic table, we're focused really on 4 things. All right. And then we'll talk about compounds here shortly. We'll define compounds, molecules, elements, all in just a few minutes. So this periodic chart is nice. It's got the color coding to show us which elements are the major elements of the body, and those four major elements: hydrogen, carbon, nitrogen, and oxygen. So H, C, N, and O, those are the four that make up the majority of all of everything in the body. When we look at a protein, it's going to be made of those four items. When we look at a carbohydrate, it's going to be made of three out of those four. When we look at a lipid or a fat or a steroid or a hormone, it's going to be made out of three or four of these. So almost everything in the body is what we call organic compounds, are going to be hydrogen, carbon, nitrogen, and oxygen. So H, C, N, and O. Those are the four you're going to see as a theme, kind of this semester, but definitely next semester in advanced. These lighter colored ones are trace elements, so we do certainly have a handful of trace elements. A couple of these other pink ones we'll look at, again, more detail next semester in advance, but Na is sodium, okay, and then Cl is chloride or chlorine, okay, calcium, magnesium, potassium. Okay, so this little batch over here of these four, these are items that, again, we need to consume in our diets: phosphorus, sulfur, and chlorine. Okay, so those are the big ones. All those other blue ones, we don't have to worry about at all. I'm going to skip. This slide's not, again, we just looked at that. What are the important ones? So in black and white: oxygen, hydrogen, and carbon. Again, you could throw nitrogen in there as well. These are found in the cytoplasm. What is cytoplasm? Remember, that's the cell fluid, the fluid inside of a cell. Well, maybe you don't remember because we haven't done a heck of a lot with cells yet, but that's what that is: cell fluid. So inside of a cell, we're going to see a lot of CH and O. Okay, atoms, when we look at atoms, we're going to look at the smallest structure of the element. So the elements, so when we go back to our periodic table, we're going to see a bunch of numbers and letters. So the atomic number above the letter, that's what we call the atomic number. That is going to be the number of protons in the nucleus of the atom. Okay, so their helium is going to have 2 protons. That atomic number, that number of protons, is also the same number of electrons. And electrons are going to be what float around and kind of orbit the center or the nucleus of the atom. Okay. So let's go back to, again, I'm not here to get you guys too wound up about some of this, but I think most of you have heard these terms before. Protons and neutrons make up the center or the mass of the atom. Okay, the atomic mass is protons and neutrons. The number of protons does not change for a particular element. Okay. However, the number of neutrons can change, and we'll look at that shortly. We call that an isotope. Okay. An isotope, the element is the same, but we've got a slightly different number of neutrons. So we could have isotopes of the same element. So we'll look at that again, not to get you too freaked out. Okay, protons and neutrons, those are in the center. They make up the mass. Electrons are what orbit around the center. Electrons are extremely lightweight. Okay? Electron number happens to be the same as the proton number. So if we go back and we look at carbon, for instance, carbon's atomic number is 6. What 6 tells us is that there are 6 protons in the nucleus, okay, and 6 electrons orbiting around that nucleus. Okay, so 6 protons, 6 electrons. We look at the atomic mass and we see 12 and 12.02. Well, remember, atomic mass is protons plus neutrons. Remember, if protons are 6 and the atomic mass is 12, that means the number of neutrons must also be 6. Okay, so 12 is 6 plus 6. Okay, so for carbon, you have 6 protons, 6 neutrons, 6 electrons orbiting that nucleus. Now, again, we can have isotopes. We could have 7 protons or 7 neutrons with those 6 protons, and that would give us an isotope of carbon. Okay, so if we had 7 protons, we now have a new element. 7 protons would mean we have nitrogen. So number of protons tells you the atomic number. The atomic number tells you number of protons. So that's the same thing. That also tells you the number of electrons. So if you were asked how many electrons does the element chlorine have, look at the atomic number is 17. That tells us how many protons, which is 17. That also tells us how many electrons, which is then 17. I could ask you one you've never heard of, lead, PB. How many electrons does PB have? 82. Okay. How many electrons does RA have? 88. How about fluorine? 9. Okay. So electrons, protons, same number. So we could even say protons and electrons. Okay, it's the same number. Neutrons can change. Remember, atomic mass is neutrons and protons combined. Okay. It's showing a couple different models. Again, we can figure out what element this is just counting 1, 2, 3, 4, 5, 6 electrons. So we're going to have 1, 2, 3, 4, 5. The 6th proton is going out the back. You can't see it. It's clogged. That's why for this particular element, we'd want to rely on the electron number, because in 2 dimensions, think of this one right here. This proton is coming out at you. And then opposite behind it, there's a proton going away from you. So there are going to be 6. So we go back to our periodic table, and we would see that this is would be carbon. It's an atom of carbon. This one too, this is just another way of writing it out. Okay, so when we look at properties of atoms, again, that atomic number of protons, which will then tell us number of electrons. The mass number is the number. Mass means there's some sort of structural weight to this. There's density to this, and remember, the electrons are kind of floating around the outside, whereas the nucleus, that's where the weight is. So the nucleus is where the mass number, which is where the protons and neutrons are located. We already know the number of protons is 6, for instance, for carbon. If the mass number is 12, then we could deduce that the number of neutrons then is also 6. But if the mass number is 12.2, that means I don't have more protons. That's always going to be 6. That means the neutrons have gone up. Okay, those energy levels, that's where the electrons live. Think of electrons as electricity, as energy, okay, energy shells. Okay, we have what we call the rule of 8. That's octet rule. So each of these elements is going to have what we call an electron cloud kind of around it. And that electron cloud is going to have multiple layers depending on how many electrons we have. The first layer of the electron cloud is only going to have 2 maximum electrons. After that, subsequent layers can have up to 8. And that rule of 8 or octet rule comes into play when we look at bonding of these elements. Okay, so we'll keep that in mind. 8, again, not hugely important, just slightly. Okay, so what's a molecule? A molecule is 2 or more atoms bonded together. So if I take 2 atoms of oxygen or 2 oxygens and I bind them together, and I bind them together, I have O2 or oxygen. So you're breathing oxygen molecules. You're not breathing oxygen, just element or an atom of oxygen. You're breathing 2 bonded together. So you're breathing in 2 oxygens. That's called a molecule. A compound is when we take 2 or more elements and bind them together or bond them together. Okay, 2 or more elements. So for instance, a compound would be sodium and chloride, okay? NaCl. Okay, carbon dioxide. Okay, so I've got a molecule of oxygen and I bring in a carbon, which is a different element. Now I have CO2. That's going to be a compound. Okay, and then we have what are called ions. Okay, you can see that at the bottom. Ions are electrically charged atoms or molecules. And electrically charged means it's going to have a positive charge or a negative charge. And what this means, and we'll see it in a little more detail, is some of these elements can donate one of their electrons or multiples, but we'll just stick with one. Some of these elements can donate an electron. And if they do, they become a positive ion. Okay? Some of these elements are lacking an electron, so they're going to receive, they're electron receivers. Okay? They would have then a negative charge. They were lacking, so they were negative. So and typically that's where that octet rule comes in. We'll usually see positive ions come from the left side of the periodic table. Positive ions come from over here. These can donate 1 electron. Na, sodium, has 1 electron in its outermost shell, so it can donate it. And then it becomes Na plus, becomes a sodium ion. Potassium, same thing, has 1 in its outer shell. It can donate that 1 electron and it becomes a positive ion. Magnesium and calcium have 2 floating in their outermost shells. So magnesium can become a +2, meaning it donated 2 or had 2 to donate and donated them. Calcium has 2 extras in its outer shell, so it can donate them. And again, where do they usually go? Usually somewhere over here. Okay, Cl. Okay. These have 7 in this column here. They're going to have 7 electrons in their outer shell. So they're going to be, they want to get to 8 so they can, they're ready to accept. Okay. So this is where we're going to find our negative ions typically. Okay, I've probably said too much already regarding chemistry, so let's keep the line moving. Isotopes, again, an isotope is when I can alter the number of neutrons in the nucleus. So hydrogen, just as an element, okay. And deuterium, I can change the number. It's still hydrogen. Now I have 2. Tritium, it's still hydrogen, but notice this is the neutron. I can change the number of neutrons again. Okay. We can see the same thing with carbon. Okay. We have isotopes of carbon. Just remember, an isotope is all the ability to alter or it's not the ability. How do we get an isotope? We've added a neutron. Okay, protons and electrons cannot change. Okay, otherwise the element changes. However, the number of neutrons can change, and the element still stays the same. We call it an isotope of that element. Okay, chemical bonds is the next stop on the tour. We're just going to look at a couple of different chemical bonds. We're going to look at ionic or electrovalent bonds. Ionic or electrovalent. Okay, let's just look at the word ionic for a second. Ionic, so we're dealing with ions, and remember, ions either donated or gained an electron. So an ion is either going to be a positive charge or a negative charge. If it's positive charge, it means it has an electron that it donated. If it's negative charge, it means it received an electron. When we look at sodium and chloride, we see the donating of electrons occurring. That's an ionic bond. So two ions, a positive ion donating, okay, or donating one of its electrons. Okay. So if two ions, a positively charged and a negatively charged, come together, that's called an ionic bond. So when we have a Na plus coming together with a Cl minus, okay, we have sodium chloride. That's an ionic bond. Okay. Valent means the outermost shell of any of these elements. So when we talk about electrons and that outer shell or that electron or those electron levels or clouds, the outermost shell, which is the only shell where I can donate or receive electrons, we call it the valence shell. So electrovalent. So there's electrical charge, electrons in the valence shell being donated and received. Okay, a covalent, co means together or cooperate. So covalent bonds share electrons. They don't donate. Okay, so we'll look at those in a couple of examples. So when we look at a sodium atom, we can see that there's an extra electron in its valence shell. Remember the rules of 8. So we have 2, 4, 6, 8 in this second shell. Only 1 in the outer shell. We look at the chlorine atom and we have 1, 2, 3, 4, 5, 6, 7 in its outer shell. So remember rules of 8. Chlorine is 1 away from being at 8. Sodium, if I remove this, is now going to have 8 in its outer shell. So this is no longer its outer shell. If I remove this electron, which we see down here, I remove the electron, I still have the same amount of protons, so I still have sodium, but I lost an electron. I donated that electron over, so now I have to denote that it's a sodium ion. It's no longer the sodium atom or the element sodium. It's now a sodium ion. Okay, it donated. It's happy because it donated. It's positive because it feels good about donating. Chloride ion had 7 in its outer shell. It just received the 8th. So now it's happy. It's complete. Notice again, we did not change the number of protons, so it's still chlorine. But instead of a chlorine atom, we now have what we call a chloride ion. It's a negative charge. It just means it was lacking. It was in the negative, but we gave it more negativity, right? So electrons, remember, are negative. So we gave it that extra negativity that it needed. That's why we denote it as having that negative charge, because it has an extra negative now. This one lost some of its negativity, so now it's positive. Hey, and at this point, once we've removed that and donated it, now we have complete outer shells. We have an 8 over here, we have an 8 over here, and that ionic bond's going to form now because of the charges are opposite. We have a positively charged ion and a negatively charged ion. Just like magnets, they're going to come together and bond in what we call an ionic bond. So again, ionic bonds are when we donate electrons. That's going to make the donor more positive. It's going to make the receiver more negative, because remember, electrons are negative. So if I donate a negative, the one getting the negative is going to be more negative. And because I lost some negativity, I'm more positively charged at that point. Still the same element, because again, we didn't alter the number of protons in the nucleus. Okay, covalent bond. I've got a hydrogen atom. Hydrogen is a 1, so that means it has one proton, one electron. So I take one hydrogen, I put them together. And remember, the rule of 8s is for the second shell on out. The first shell is a rule of 2. It's not really a rule, but we'll call it the rule of 2. Okay. So I can have a maximum of 2 in that inner shell. So these guys can share. And then it becomes a hydrogen molecule. Okay. See the same thing with oxygen. Oxygen can become an oxygen molecule, or O2. Okay. Put carbon in between it. Okay. Carbon is a atomic number is 6. So that means it's going to have 2 electrons in that first shell, and then its outer shell is going to have 4. Okay. And then each oxygen has 6 in their outer shell. So 6 can combine with 2. The other 6 can combine with 2, and that gives us our total of 8. So these guys are sharing electron shells. So 1, 2, 3, 4, 5, 6, 7, 8. Okay, so oxygen is complete. We look at carbon, 1, 2, 3, 4, 5, 6, 7, 8. So carbon is complete. And then oxygen we know is complete, 4, 5, 6, 7, 8. And how did we get these 2? So remember, oxygen had 6 in its outer shell, so it only needed 2. This oxygen had 6 and it needed 2. Well, carbon has 4. So it could share 2 with this guy to get to 8. It could share the other 2 with this guy to get to 8. And then they can all be happy. And we call that a double bond and a double bond. And that's carbon dioxide, CO2. Okay, so covalent just means we're sharing electrons. Ionic means we're donating. Hydrogen bonds are next. We see these are a little off the beaten path. These are the weakest bonds. We generally see this with one hydrogen connecting to either an O, an N, or an F. So hydrogen connecting to O, N, or F. And then another hydrogen bonded to the O, N, or F. So we could have 2 hydrogens bonded with an O. Okay. For instance, that gives us H2O. So remember, water has a 6 in its outer shell. So it needs 2 more to be complete. So these are almost like covalent bonds, right? Electron sharing. Hydrogens, remember, have 1 each. So if one hydrogen comes in, now oxygen's at 7. Well, let's bring another hydrogen in and make it an even 8. So now the oxygen gets satisfied. It has its 8. Hydrogen is satisfied because it has 2. Remember, hydrogen only needs 2 to be happy. Okay. And then we have negative, positive, positive. These are poles. So this negative pole of this hydrogen bonded H2O molecule can receive another positive. This can receive a negative. This can receive a negative. So we can start connecting all of these water molecules to form water. And these are hydrogen. And again, these are easy bonds to break. We know this with water, right? We can break these bonds. We can heat up the water and break them, get steam out of the deal. We can cool the water excessively and get those bonds to get real tight and turn water into ice. Yeah. Hey, chemical reactions, just a quick pit stop here. There are four basic types. I just want to mention two: synthesis and decomposition. All this means is we can either build things up or synthesize. Will see these terms again. That's why I'm bringing them up even just a little bit. We'll see them way again when we get to advanced, but you'll see them next week when we talk about cells, and we'll see them a little bit throughout. But it's synthesis, that just means we're building things up. So your cells are what are doing this. Your cells are synthesizing or building things up. So we'll take one strain of amino acids and another string of amino acids, and we'll build them, and we'll form a larger protein, or maybe a muscle or a hair follicle or an enzyme. Decompositions, just like it sounds, we're breaking something apart. So digestive enzymes or any type of enzyme participates in decomposition. We also call these catabolism or catalyzing. This we call anabolism or anabolizing. So catabolism, okay, catabolism is decomposing, breaking something down. And usually we see the word lysis, or the suffix lysis, and oftentimes hydro, which means water. So water added to something will break it apart. Okay, that's what catabolism means. Usually, again, in the presence of enzymes, which we'll talk about in a few minutes. Anabolism is a fancy word for synthesis. Again, we're building things together. And this we usually see condensation or water. We see this as what we call a dehydration. Okay, dehydration. So hydrolysis means we add water. Dehydration means water is leaving. Again, total surface level stuff with this. I don't want, it's probably gone too deep as it is, but I don't think so. I think we're doing just fine. And we're almost there. We've almost made it to the end. Organic and inorganic compounds. Okay, organic compounds are going to have what we call carbon or carbon hydrogen bonds. Okay, so organic. In order for a molecule to be organic, it has to have a carbon and a carbon bonded together, so at least two carbons, or a carbon and a hydrogen. So how about carbon dioxide? CO2. Okay, CO2. You have one C and two oxygens. One C, two oxygens. Okay, that is not organic. Okay, that would fall in the inorganic category. Most inorganic compounds do not have carbon, but there are some, right? Like carbon dioxide, for instance. Okay, organic molecules. Here are the four biggies: carbohydrates, lipids, proteins, and nucleic acids. Okay, those are the big four organic molecules. Okay, we'll look at some inorganic compounds here in just a second. Okay, carbs, fats, proteins, and nucleic acids. Now, in the outline, I'm not going to go through too much of in the slides, but in the outline, I give you a couple kind of examples and a little bit of discussion on carbs, lipids, proteins, and nucleic acids. So I'm going to do that here, really just to introduce you to these, primarily the first three terms there. I know all of you have heard of carbohydrates, and I'm sure I know you've all heard of proteins too. You've maybe not heard of lipids, but you have heard of fat. So fats and lipids go together. They're the same thing. So carbohydrates, lipids, and fat, or carbohydrates, lipids, and proteins, okay? Or carbohydrates, fats, and proteins. Carbohydrates are sugars. Generally, we would see a carbohydrate end in OSE, so like glucose or fructose, lactose. Okay, so carbs generally end with OSE. We have what are called complex carbohydrates, and we have what are called simple carbohydrates. Okay. Complex carbohydrates are going to be long chains of carbons. Remember, all of these are going to have carbon probably carbon and carbon and or carbon and hydrogen. Every single one of these, remember, because they're organic. Carbohydrate, even the name carbo, carbon, hydrate is H2O. So if we put CH2O together, that's what the ratio is for a carbohydrate. Okay, carbohydrate. And again, these are going to be your simple sugars and your complex carbohydrates. We see the word starch with complex carbohydrates. Okay. You'll oftentimes see saccharide in there. Like saccharide means sugar. So you'll see monosaccharide, disaccharide, polysaccharide. That just means single chain sugars, double chain sugars, and many long chain sugars. So again, simple carbs, complex carbs, also known as polysaccharides, also known as starches. So these are all terms you've maybe heard at some point, but maybe didn't associate them with carbohydrates or sugars. Okay. Why do you need carbs? You need carbs for energy primarily. These are it's the kind of the ingredient that cells use to make energy, or not really make it, they convert energy. Okay. Remember, energy cannot be produced or destroyed. You can only cycle it and convert it and break it down and convert it again. So carbohydrates, that's your energy molecule, or what leads to the energy molecule. Okay, lipids are fats. We would see glyceride instead of saccharide. We'd see glyceride, so like triglycerides. Okay. Lipids, you maybe heard of like cholesterol, HDL and LDL. Those are cholesterol levels. Okay. Steroids are cholesterol based. Some of our hormones are lipid based or fat based, like testosterone and estrogen. Those are going to be made of lipids. And then proteins, those are going to be what give you structure. Those are what give you enzymes, which we'll talk about in a few minutes. Proteins are made from amino acids. So the building blocks of proteins are called amino acids. And again, you've already heard of amino acid at some point in your life. A lot of people who work out, or they drink protein drinks, and helps to regenerate and with some of the amino acids and the building up of the structures. I'd say post-workout protein is better than pre-workout. Generally, you need energy. So usually honey and water is going to be your best energy source. It's your fastest energy. Honey has the right kind of carbohydrates in it. It helps to repair tissue even while you're working out. And then proteins are good, for maybe post-workout. But yeah, wolfing down a big protein bar and then guzzling a protein drink and then pumping iron and, that's proteins sit in the guts. It takes 4 to 6 hours to move proteins out of there. They're great for building tissue. They're not your body's preferred energy source. In fact, they're number 3. Eesh. Don't believe that keto diets and all that are good for you. Proteins are the least preferred source of energy. Lipids are second least, and carbohydrates are number 1. The reason carbs get a bad reputation is because of the type of carbohydrate that people consume makes them sluggish, makes them maybe obese. It could potentially give them diabetes issues, metabolism issues. That we're talking about like soda pop, we're talking about, buckets of gummy bears and, candy bars and, that type of milk, a lot of too much dairy, that's full of sugars that can really blimp you up. Um, so again, it's the body's preferred energy source is fruits and vegetables, honey, starches, pastas. Those are the carbs that you need. If you're going to do workouts, before you do the workout, you need to take in energy in the form of carbohydrates, and not complex carbs though. Don't eat a bowl of pasta while you're running on the treadmill. Oof. So, by the way, too, working out is stress, right? You're inducing stress. You're increasing blood pressure, increasing respiration, um, body temperatures. All these things are happening, right? So exercise is a form of stress. It's good stress, but it's still stress. One thing that happens during stress is that your digestive system gets kind of squeezed out. So you're not really thinking about eating and consuming things when you're in the thick of working out. So um, anyway, so beforehand, you want to keep it light on the guts. You'll find that you'll be able to sustain your workouts longer with just, I'm telling you, if you take a 20-ounce bottle of water and you pour about a tablespoon or more of honey in there and shake that up and guzzle that, maybe eat an apple or something, or some orange slices or some grapes, and you'll have that long sustained energy. And then while you're working out, keep drinking that honey water. Okay. So don't be wolfing down protein bars right before you want to go, take a tour of on the treadmill, or whatever, Bowflex. I don't know what people use now. I don't even know if that exists. So anyway, organic and inorganic, right? Organic, it's going to be carbon-carbon or carbon-hydrogen bonds. If we don't have carbon-carbon bonds, carbon-hydrogen bonds. Oh, by the way, nucleic acids, that's going to be RNA and DNA, which we spend a lot of time on that next semester. So I'm not going to get too deep into DNA and RNA right now. Okay. Again, you've all heard of DNA, I'm sure, at some point. Comes from the nucleus, right? The DNA. So next week when we do cells, we'll circle back and look at the nucleus and DNA a little bit more. All right, so the big inorganics, we've got oxygen and carbon dioxide. Again, these are the kind of the opposites. We want the oxygen is the grocery coming in, carbon dioxide is the waste that leaves. Okay, so those are a couple of your two biggies on the inorganic end. And then, of course, all of those electrolytes. You've all heard, you guzzle your Gatorade, it's full of electrolytes. So electrolytes, we generally think of like positive ions, or just ions in general, right? Electrically charged. So any type of ion. So sodium, potassium, magnesium, zinc, chloride, and all of these ions. But the big electrolytes, again, the ones I just listed, okay? Sodium, potassium, calcium, magnesium, those are the biggies. Manganese, phosphorus, those are also good and big biggies as well. Okay, so your vitamins and minerals are going to be in that inorganic group. Okay, water. Why do you need hydration? I've been talking about hydration quite a bit. Again, we looked at that polarity. Polarity means we have opposite charges, opposite ends. So when we looked at that water molecule, we saw that polarity. Oops, there we go. We've got positive end here, negative end here. That's what polarity is. So the polarity of that molecule is very important. It becomes a solvent, not just for other water molecules, but any type of positively charged or negatively charged molecule or ion can latch on to water. It has a high specific heat. That simply means that it takes a high temperature to get it boiling. We have to get all the way up to 100 degrees Celsius to get water boiling, which is 212 degrees Fahrenheit. So thankfully, we're not exposed to 212-degree Fahrenheit temperatures. So that high specific heat does allow us to maintain a consistent and constant temperature. The high heat of vaporization simply means that excess heat can vaporize off of the body, because this is part of the sweating. So when you sweat, what that sweat does, those water molecules that are forming on the body actually are going to evaporate into the air. And as they evaporate, they're pulling that heat with them. So that's how we call it evaporative cooling. So we're going to look at that as well on Thursday, your case study. So your case study discusses a little bit of homeostasis, positive feedback, negative feedback. It discusses our set points and stimulus in, response out. It discusses, we'll get into properties of water and hydration a little bit. We'll talk about evaporative cooling and heat, high heat of vaporization. So vaporization just means it's able to evaporate. So, so as we get hotter and are sweating and working out, again, that water is going to pull the heat with it as it evaporates off of the skin. A couple I just threw 5 more on here as well that we see. Most abundant compound, 65% of your body is water. Roughly 65% of the planet happens to be water as well. It's the insides of our cells are water. Around our cells are water. In fact, the bulk of our body, right, is 65. About 2/3 of our body is just water. A majority of that amount of water is inside of our cells. So most of the water is what we call intracellular or inside of your cells. We call it cytoplasm as well. So anyway, so we can transport a lot of stuff. The blood, right? Your blood is about 55% water. So we call it, again, plasma. That's for transporting chemicals. It's a lubricant for our joints. And lastly, the last thing on the docket today is enzymes. Enzymes are going to be protein-based. These are chemicals in the body. In essence, as we go through this semester, next semester, we're going to encounter a few different words that basically mean chemical. So enzyme is one of those words. An enzyme is a type of chemical in the body. Other types of chemicals in the body are going to be hormones. You've all heard of hormones. Okay? Hormones and enzymes. Those are 2 of the primary chemical groups that we discuss really throughout this semester, next semester, and pharmacology, microbiology. You will discuss enzymes, okay, as a type of chemical. Kind of an umbrella term for multiple types of chemicals. Enzymes. Okay. Hormones. Okay. There's another one we'll see shortly called neurotransmitters. Okay. You've probably heard of neurotransmitters, but maybe didn't, like, specific ones. You've maybe heard of serotonin. You've maybe heard of dopamine. Okay. These are endorphins. Okay. Serotonin, endorphins, multiple types of neurotransmitters. Dopamine is the neurotransmitter in the brain that affects people with Parkinson's disease. Okay. Dopamine. So enzymes, hormones, neurotransmitters. Those are the 3 main classes of chemicals. For now, we're just going to talk about enzymes. Enzymes are protein-based, meaning that they're primarily composed of amino acids. And when we see a word written out, if it has ASE at the end, it's going to be an enzyme. Okay. I could give you a 25-letter word, and the last three letters are ASE. And I could ask you, this long word an example of an enzyme, a neurotransmitter, a hormone, or an elephant? You're all going to say it's an enzyme. You've never even seen or heard of this long word, but you look at those last three letters and see ASE, boom, it's an enzyme. What does an enzyme do? It speeds up a chemical reaction. It's what we call a catalyst. Okay? So it catalyzes or speeds up a chemical reaction. Okay? A lot of enzymes are going to be located in the digestive tract or within the digestive system. A lot of other enzymes are going to be located inside of cells. A lot of action is going to take place inside of cells when it comes to chemical reactions. Okay? Most of these are, again, going to be starting with breaking things down that we consume. The bloodstream takes those tiny microbits, delivers all those microbits to the cells, and the cells can continue the process by adding more enzymes and breaking these chunks into even smaller bits. So if I were to eat a chunk of steak, just a little chunk of steak, chew it up good, swallow it, goes into my stomach, right? If I didn't have enzymes, that chunk of steak is likely going to take a really long time, days, weeks, maybe months to break down. So if I take that piece of steak and I just add water to it, is it going to dissolve? Is it going to break up? Maybe a little, but not much. Now, if I take an enzyme, what we call a protease, for instance, it's a type of enzyme. Okay. We'll see one called, we'll see another one later on in digestion next semester that I could add to a protein, and it'll almost immediately dissolve it into tiny, tiny amino acids. Okay, so enzymes speed up a chemical reaction. They enable us to take these big chunks of food that we consume, whether it, steak and potatoes and bananas and, green beans and butter. And oh man, it's lunchtime, right? I'm going to wrap things up real quick here. Okay, we eat all that stuff. It goes into the body. Yeah, we poop at some point, but the poop isn't the chunks of food we ate. At least I hope not. Hopefully all that food you ate got broken down by enzymes and all the little particles, what we call amino acids and simple sugars and fatty acids, like omega-3, omega-6, right? You guys have heard of fatty acids. That's what we get from the breakdown of fats. So enzymes, even though they're protein-based chemicals, still will enable a carbohydrate or a lipid and, of course, a protein to break down a lot faster and in a lot smaller bits. Lactase, I put the amylase, by the way, that's a carbohydrate one. Lactase is the one that breaks down lactose. So when I consume lactose and, ice cream, hey, yeah, big bowl of ice cream, and oh man, how am I going to get that? And lactose is a double. It's a what we call a disaccharide. So I add an enzyme called called amylase, or we can get even more. It's more specific, lactase, and that lactase enzyme will take lactose and it'll break it into galactose and another simple sugar. Okay. If you don't have lactase, then lactose won't get broken up. It'll give you gut pains and all kinds of stuff. So people can take supplements if they really need their ice cream, which we all do. Okay. So anyway, those are the different types of enzymes. Okay. And again, all of this